Well, hello to the most beautiful and amazing people on the planet. I'm Selma and I'm just trying to help. Basically, today we'll begin talking about the histology and we're going to begin with the CNS module, okay, the neurons and stuff. Basically, today we're going to be discussing the nervous tissue. What is the nervous tissue all about? We'll begin talking in the nervous tissue about the neurons, the nerve cells. Okay. Well, the first thing you should know about the nerve cells or the neurons, that these babes actually are the structural and functional unit. What does that mean? I mean, the neurons all together, they form the central nervous system. I mean, a lot of axons together make a nerve. Okay, and they're the functional unit as these babes are the one who does the physiological function, which is the transmission of the impulses and the signals to the CNS or away from the CNS or in between the neurons, okay? So basically, these are the structural, I mean a lot of neurons together, they form the CNS, okay? And the functional unit as well, as they do the physiological function, which is the transmission of the impulses, baby. Okay, well, if we want to talk about the neurons, each neuron consists of two parts. Okay, the first part here is the cell body. What is the cell body? Well, they like to call it perikaryon. And I need you to have, let me write it down, carry-on. I need you to have a very nice moment and with yourself to think about the name and understand it. What does peri mean? Peri means around. What does karyon mean? You remember the word karyotyping? Yeah, karyo means nucleus. And karyotyping, as a background information, was a test for like the DNA. You want to see the chromosomes all together. And this is more details, don't give a shit about it. But I mean, the peri means around, karyon means nucleus, which means that the cell body is surrounding the nucleus. This is why they call it the perikaryon, which is the cell body. Okay, and we're going to discuss the shape and stuff, all about it. And the second part here, let me get the color back, which are the processes. When we have two processes in here, we have either the axon, that big one, okay, or the dendrites. Axon take the impulses away from the cell body, and the dendrites, they take the impulses to the cell body. Is that okay? So a neuron basically consists of two parts. It's either a cell body, and we call it perikaryon. What does perikaryon mean? Baby, perikaryon. Peri means around, karyon means nucleus. So it's around the nucleus. What about the other part? Oh, it's the processes. What are these processes all about? I mean, axon takes it away from the cell body, and the dendrites, they take impulses to the cell body. Oh, that's perfect. We'll begin with the cell body, which is called perikaryon. I just explained that a moment ago, which peri means around, karyon means nucleus, baby. Okay, let's go to the next slide. And we want to talk about the size of the cell body, which is basically not the same. It varies. I mean, from 4 micrometers, okay, to about 100 micrometers. And where's that and where's that? I mean, it's 4 micrometers. It's something called the granular cells. Where are those granular cells? Granular, blah, blah, blah cells. Okay, these ones are in something called the cerebellar cortex. You know, the CNS consists of cerebrum, cerebellum, so cerebellar cortex, the cortex of the cerebellum, yeah. So it's about 4 micrometers in the granular cells of the cerebellar cortex. And they may be so huge. Where would they be huge? I mean, some were called the motor neurons. The neurons, motor neurons. The neurons responsible for your actions of, like, I mean, if you want to like flex your biceps or something, these are the motor neurons, okay? They are responsible for the movement and stuff. And I do not want to go into further details because this is just histology, but whatsoever. Motor neurons, and these motor neurons are present in the spinal cord. So hey, well, let's go to the most important part over here. How can I memorize where is it 4 micrometers and where is it 100 micrometers? Yeah, that's so easy. Just take the mnemonic with me. 100 has 2 zeros and motor has 2 O's. So 100 and motor. This is the most important here. So remember the one with the 2 zeros and the one with the 2 O's. Okay? Consider this a 0 whatsoever. So 4 micrometers in the cerebellar cortex, the granular cells of it. And 100 micrometers in the motor neurons because it has 2 O's of the spinal cord. Okay, this is actually very nice. And then we want to talk about the shape. Well, the shape can be unipolar, bipolar, or multipolar. Well, how would I differentiate? Well, if it was unipolar, and we'll talk about it later, we'll know that the unipolar is a T-shaped and it has only one process whatsoever. Let me not talk about it. But the most important thing that you need to know about the unipolar is that it has a U. Here's the U. And it's in the globular cells with a U. Okay, is that okay? So what you need to know, that the unipolar cells with the U are in the globular cells with also the U. And then the bipolar, bi means two, and it's fusiform in shape. Okay, what shape is it? It's fusiform. And fusiform has two F's, and bi means two. So how to remember? First of all, unipolar with a U, globular with a U. And then the bipolar cells, the shape, has two F's, so it's fusiform. And the multipolar, oh, it's stellate. You know what the stellate means? Stellate means it looks like a star. So it's in the stellate and pyramidal cells. I mean, the multipolar, it's stellate, something called pyramidal, and something called pyriform. You know, pyriform, the, here. Let me get the green one. Pyriform means it looks like a pear. Yeah, let me get the pen over here. This is pyriform. Okay, so the multipolar is stellate. I just wrote stellate because it looks like a star, and the star has a lot of angles, as you can see here. It can also look pyriform or pyramidal. That's the pyramid, baby. Okay, so again, I need you to say with me, unipolar is in the globular cells. Bipolar is fusiform because it has two F's, and bi means two. And the multipolar is stellate because the star has a lot of angles. It can also look pyriform or pyramidal. What can it look like? Pyriform or what? Pyramidal. Excellent. Let's go to the next slide. Now we want to talk about the structure of the cell body itself. I know this is very frustrating of me. What are you talking about? What is this drawing? This is the cell body. These are the dendrites, and this is the axon. This is something called terminal alborizations. Don't give a shit about it. But let's talk just about the cell body. First of all, we will divide the cell body. It's perikaryon. So it's either a nucleus or what's around the nucleus. What is around the nucleus? It's cytoplasm. So we'll divide it into two. Either nucleus or cytoplasm. What do we begin with? We begin with the main, baby. It's called perikaryon. And so then who's the main here? It's the nucleus. So we'll begin talking about the nucleus. That I can see here that the nucleus is large. Look with me. Let me get a cute color that I did not use. Oh my god, I used all the colors. Look, the nucleus here is large. I can see the nucleolus very well. Of course, oh my god, here's the nucleolus, here's the nucleus. I know the nucleolus should be pale. I did not have the colors. Okay. I can also see that it's spherical. Yeah, and I can see that the cell body is basically surrounding it, which means I'm not a liar. It's still perikaryon, baby. So I can see that the cell body is surrounding it, and it's spherical. It's huge. It has a prominent nucleolus. Oh, and it's pale. Do you see it dark? Can you see it dark? No. So it's pale. And any nucleus that is pale, this means that it's very active. Right? Because the DNA is not condensed. It's open. It's synthesizing. It's working, baby. So this cell is pretty much very active. Okay, why is that? Because it's synthesizing the neurotransmitters. So it's pretty much very active. So I want to know, again, the nucleus, oh, it's spherical, it's pale. Oh, really, the cell body is surrounding it. Oh, they didn't call it. They did call it perikaryon for a reason. Beside it's large, okay? Right, I can see a prominent nucleolus, and it's pretty much so cool. Let's now forget about the nucleus. What if you want to talk about the cytoplasm? Well, cytoplasm has three things. I need you to look with me. Cytoplasm has three things. One, it has organelles. This is absolute common sense, baby. What about number two? It has a cytoskeleton. The filaments and the tubules, microtubules and stuff. And the last thing, it has the inclusions. Yeah, why isn't it writing down inclusions? Yeah. So what does the cytoplasm have? Oh, it has a cytoskeleton, has some organelles, and it has some inclusions. What are we going to begin talking about? We'll begin with the organelles. Let's take a look over here. First of all, I can see that it has here a rough endoplasmic reticulum. I know you want to tell me, hey, you're very bad at drawing, but this, oh, I'm sorry, it's an RER. Because believe me or not, this is a rough endoplasmic reticulum, but I'm very bad at drawing. And these are free ribosomes. Why is that? Because this cell is pretty much very active and it synthesizes a lot of things, right? So neurotransmitters and stuff. So it has highly intense synthetic activity, which means this is a very, very well developed rough endoplasmic reticulum. And beside it has free ribosomes. Why is that? Because it's the function, babe. How can we have a highly developed rough endoplasmic reticulum without those ribosomes? So again, what are the organelles? One, it has a very, very highly developed rough endoplasmic reticulum with a lot of free ribosomes here. Free ribosomes. I made sure with the color, baby, they're matching. And this is a rough endoplasmic reticulum. Let me remove this shit that I'm drawing. What about number 2? Well, I can see here there's a Golgi apparatus. Yeah, believe me or not, it's a Golgi. And here it's another Golgi, but it's pretty much small. And I can see that they are perinuclear. And one thing, well, you remember from your cytology lectures, they said that the Golgi apparatus or the Golgi body is either perinuclear in the nerve cells or apical in the secretory cells. You remember that? No, doesn't matter. I mean here it's perinuclear. It's around the nucleus. Okay, because here, baby, the nucleus is very, very important, so it's the main bulk. So again, the nucleus is spherical, large, pale. It has a prominent nucleolus, and it's in the middle. Okay, it's surrounded by the Golgi apparatus. Even the cell body itself was called perikaryon. Great. What about, and also here the cytoplasm, of course it's basophilic, and it has highly developed RER and a lot of free ribosomes because it's active, baby. Why isn't this shit being removed? My God. Anyway, whatsoever. Okay. I cannot erase this thing, but it's okay. And then we do have a lot of mitochondria. Can you see the mitochondria over here? This is mitochondria, which means it's active. Why would we need the mitochondria very scattered in the cytoplasm right like that? Well, because it's highly active. Do not ask the same question again. Okay, so it's highly active, has a lot of mitochondria scattered in the cytoplasm. It has a perinuclear Golgi apparatus, highly developed RER, a lot of free ribosomes. Say with me, I caught you not saying. And now I want to ask something for these smart ones. Well, take a minute to think about it. Do you think the nerve cells would have any centrioles? You have 5 seconds to think about it. 5, 4, 3, 2, and 1, and absolutely not. Why is that? Because nerve cells cannot divide, baby. Can you see a nerve cell? If the nerve cell is dead, then it's dead, baby. It cannot divide. Okay. So, nerve cells actually cannot divide, so definitely they would have no centrioles. Okay, now we're done with the organelles. Can you say them quickly with me? Yeah, of course. First of all, we have a very highly developed rough endoplasmic reticulum. Have a lot of free ribosomes, mitochondria scattered throughout the cytoplasm, which is absolutely basophilic. Don't forget, there's something called the nestle body. Oh, I forgot to say it. Oh, thank God I saw it. This is called the nestle buddies. The number of them varies from one neuron to another according to the type and function. Okay, and these are very, very important because the nestle bodies or the nestle granules for the synthesizing. Okay, because it's highly active. Okay, so these are the nestle bodies. What else? We have the Golgi apparatus, perinuclear, very much important for the secretory stuff. And I want to remove everything I drew. Don't forget, highly developed RER, and of course, mitochondria scattered throughout the cytoplasm. Don't forget the nestle bodies. Is that okay? Yeah, okay. It has any centrioles? No centrioles here, baby. We don't do this here. What about the cytoskeleton? Of course, I did not draw it, but we have 2 types of the cytoskeleton. One, we have the neurofilaments. And one thing I need you to know about the neurofilaments, that they provide the structural support. Okay, they provide what? The structural support. Neurofilaments are basically intermediate filaments. Okay, about 10 nanometers in diameter. One thing you need to know is that they are very, very abundant in the cell body and the processes, both of them in the neurons, I mean. Okay, and they work in providing the structural support. What about the microtubules? Oh, that's cool. Microtubules are basically for transport. Transport of what? The neurotransmitter, babe. It's been about 15 minutes. I've been saying that this cell is very, very intensely active. So it's active because it's synthesizing those neurotransmitters. So absolutely, we'll need to transport this neurotransmitter that has been synthesized. Okay, so these microtubules are arranged in parallel bundles like that, about 20 to 28 nanometers. Remember your cytology lectures. Okay, and they're arranged in parallel bundles. They help in transport of the neurotransmitter. Is that okay? Besides some other constituents like enzymes and whatsoever, but the most important thing is the neurotransmitter. So again, for the cytoskeleton, we have finished the organelles, we have finished the nucleus. Now we're in the cytoskeleton. In the cytoskeleton, okay, we have the neurofilaments, which are very small, about 10 nanometers. They're important in the structural support. They are present as fixatives, which means support, in the perikaryon, which is the cell body, and the processes, aka it's present throughout the neuron. Great. And of course, you stain them by silver. This is from the cytology lectures. What about the microtubules, which are basically from 20 to 28 nanometers in diameter? What is the importance of them? I've been saying for 16 minutes now that this cell is very highly intensely active. So it's very important in synthesizing the neurotransmitter. And who's going to transport those neurotransmitter? Yes! It's the microtubules, baby. And the last thing, we were talking about the inclusions. For the inclusions, I need an empty page. Okay, here's an empty page. This is an empty page. For the inclusions, it's me and Lily. What are you talking about? Yeah, it's me and Lily. Me for melanin. And if we go upstairs to reach here, we're going to find here the melanin as the dark brown or black pigment. You can see those here. It's the dark here. You see it dark brown? Okay, this is the melanin. Okay, it's responsible for like giving this color of your skin. You'll also find it here in the nerve cells or the neurons. I mean, they're the same thing, of course. Melanin, it's the dark brown. You'll also find me and Lily, Li for lipofuscin. The age pigment, you'll find it golden brown. Take a look over here. Let me change the font. Yeah, here. The lipofuscin, you'll find it this dark golden brown. And here's the golden and here's the brown. I couldn't mix both of them together, but they're golden brown granules. Okay, these are the age pigments from the residual bodies. What are those residual bodies? You know, when the lysosome eats its stuff, there are some residual bodies that are undigested. These undigested material are called residual bodies. And basically, they make a pigment called lipofuscin pigment. This lipofuscin pigment is called the age pigment. Okay, and the last thing, Lily, lipid granules, as you can see them over here. And this is energy reserve. What is it? Energy reserve. So again, if you want to talk about the inclusions, it's either me and Lily, excellent. Here is a me for melanin, which is dark brown or black. Okay, and it's responsible for the coloration and stuff. Okay, and it's also found in some neurons. What about Lily? L for lipofuscin, the age pigment, which is basically golden brown. And this golden brown, baby, is for the residual bodies that are undigested. What about the lipid droplets? I mean lipid. We stain lipids in Student 3, this is why I drew them in orange, but I mean, if you're using H&E, it will definitely dissolve. But whatsoever, these are for the energy reserve, babe. What is it for? The energy reserve. Okay, now we have done the perikaryon, the entire perikaryon. Let's go to the other part, where we want to talk about the processes. What are those processes? Let's go upstairs again. Let's remove everything we've drawn. Wait with me. I know I should pause the video and remove those stuff, but it's okay. We're all doing great here. Yeah. Okay, I'm not pausing the video, I can't. Anyway, this is the axon. Let's change the color, or let's highlight it whatsoever. This is the axon. Oh, this auto-detect straight highlighter. Whatsoever. And these are the dendrites. What can you find as a difference before I go and say the difference myself? Well, I can see that those dendrites are very small, they're short. This body is very long. 1. 2, I can see that those bodies are having a lot of branches. Oh, then this is number 2. Well, I can see that the angle is not definitely, it's not always a right angle. I can see this one, look. This is the axis. It's on a right angle, yeah, basically. I can see that this one originates from the axon hillock. It's the most active part. It's the most excitable part in the neuron. It's called the axon hillock whatsoever. I can see that they're branching, and you definitely cannot see that the branches will get thinner as long as the dendrites branch. The branches are getting thinner, but you would never see that here. Why? Because I couldn't draw it. How would I do it? But I mean, consider it yourself, please, kindly. Okay, so again, 1, the dendrites are numerous, there's a lot, the axon is only 1. 2, these are short, this is long. 3, basically this gets the impulses into the neuron, and this gets it out of the neuron, away from it. Okay, that's nice. What about number 4, I guess? These babies are branching, and when they're branching, they get much thinner. But that one, it's not branching, and it has the same diameter. Well, it can give some like collateral branches over here, but this is no big deal. And this gets it into, and this gets it away. Let's see, check out here. Okay, dendrites, it gets the information into the cell body. No, I want to remove the highlighter. Whatsoever. They get the information into the cell body, but the axon gets it away from the cell body. Great. What about number 2? Dendrites are numerous, the axon is single. This one is branching, and this is not branching, baby. Okay, but it can give some collateral branch at a right angle when it's like originating. Okay, what about again? Here, this one is short and this one is long. Here, yeah, the dendrites, they're here arise the axon at a right angle, but here it arises at an acute angle. Okay, acute means less than 90. Of course, I couldn't draw that too. Okay, and then as it subdivides, it gets smaller, but here it has a constant diameter. I showed it to you yourself, baby. The dendrites, I forgot to say that, it has nestles, buddies, but this one doesn't. And this one is thin, this is thick. Why is that? Because this is 1, and these are a lot. Okay. God makes everything in balance. If this is only 1, then it's thick. If these are many, so each one will be thin. Because justice, baby. So again, dendrites into the cell body, they're numerous. They're thin, they're branching, they're short. As they subdivides, they get smaller. But that one, no. It is long, it's not branching, but it can give collaterals. It is single, and it takes the information away from the cell body. Don't forget, it has a constant diameter, and it's thick. Well, if you want to classify the neurons right now, we have a lot of classifications of the neurons. And let's begin. Okay, we have according to the number of processes. Let me just, I know I'm ruining everything I drew, but whatsoever. No one gives a shit. Okay, I didn't use that color. It's according to the number of processes, or according to their function, or according to the length of the axon. I would like to begin with talking about the length of the axon. Well, it's either tall or short. Really? Yeah, exactly. I mean, if it's tall with an L, then it's type 1. And I like to say it that way. Long. To memorize it, type 1 is the long axon, type 2 is the short axon. Okay, so Golgi type 1 neurons, actually, they have a long axon. This is like the pyramidal cells in the cerebral cortex, maybe the Purkinje cells in the cerebellar cortex. Don't forget, maybe in the spinal cord, the motor neurons of the spinal cord. So again, they're either tall or short. Tall axon as the long, and the tall axon, which is the long, as in type 1. Golgi type 2, they're the short ones. Okay, where would we find type 1? I mean the pyramidal cells of the cerebral cortex, the cerebellar cortex, Purkinje cells. Don't forget motor neurons of a spinal cord. What about type 2? You'll find it in something called interneurons, the neurons that are like in the middle of a synapse, and I'll explain that more later. But I mean, these type of neurons, they do not leave the gray matter in the like the interneurons. Okay. And the neurons, like, they synapse axonically in the middle. I like to call those interneurons the third wheel, and I'll also explain it later on. Okay. And then this type 2, so it's in the interneurons, and these interneurons, they do not leave the gray matter, and you can find it in the cerebral and cerebellar cortex, and sometimes in the spinal cord. What about the function? I mean, the function is either sensory or motor or interneurons. And I said interneurons are this third wheel, baby. If you're having a couple and you're going out on a date, your friend that is on the phone with you, like, or a third wheel, is this interneurons. Okay, you can find those interneurons in the retina or in the spinal cord. Motor, when it gets out, you feel. So this is sensory, and it goes to the CNS, and then out of the CNS you have the motor neurons. Okay, which will go to the effector organ, the one that it will maybe your biceps, and it will contract, maybe to a gland, so it will secrete something. Okay. So motor neurons will control the effector organs, as the muscles, as the gland, or whatsoever, and these motor neurons are from the anterior horn cells of the spinal cord. Okay, so any anterior horn cells are the motor. Great. And then you'll find some sensory. Sensory is the one that felt, and it goes all the way to the CNS. So if it's to the CNS, it receives the sensory stimuli. I mean, I touched the fire, so I felt the fire, so it goes to the CNS, and this is all about physiology. But what you need to know here, that it is in the cells of the dorsal root ganglion. Where are they? Cells of the dorsal root ganglion. Is that okay? Okay. What if you want to talk about the number of processes now? I mean, look with me. One, you'll find the unipolar. One thing you need to know about the unipolar cells is that it has one process. It gave me a T shape. One of them will make the dendrites, and the other will make the axon. So it has a single process. The single process is divided structurally into two parts. Okay, one will make the dendrites, and the other will make the axon. Is that okay? Where can we find that? I mean, somewhere called the spinal ganglia. Well, where would you find it? The spinal ganglia. And something called mesencephalic nucleus. What is it called? Mesencephalic nucleus. And this is in the trigeminal nerve. So where would we find it? Spinal ganglia, mesencephalic nucleus of the trigeminal nerve. And then we'll talk about the bipolar. Bipolar means it has only two processes, a dendrite and an axon. What are they? A dendrite and an axon. You'll find it somewhere called the cochlear and vestibular ganglia of the ear. Cochlear, the cochlea of the ear, and the vestibule of the ear. So cochlear and vestibular ganglia of the ear. Don't forget, you'll find it in the retina and the olfactory mucosa. The retina and the olfactory mucosa. Then the multipolar cells. And I would write this down. Multipolar cells. Pyramidal, you'll find it looks pyramidal in the pyramidal cells. You'll find it pyriform in the Purkinje cells. So pyramidal and pyramidal. Pyriform in the Purkinje. So pyriform were Purkinje cells. And where are those Purkinje cells? Oh, it's in the cerebellar cortex. And where are the pyramidal cells? In the cerebral cortex. And the last thing, you'll find stellate, aka star-shaped. Where are those? Oh, in the anterior horn cells of the spinal cord. Where are the anterior horn cells of the spinal cord and the sympathetic ganglion? And that's it.